Section 9.1, the structure of DNA. By the end of this section, you will be able to describe the structure of DNA and describe how eukaryotic and prokaryotic DNA is arranged in the cell. In the 1950s, Francis Crick and James Watson worked together at the University of Cambridge, England, to determine the structure of DNA. Other scientists, such as Linus Pauling and Maurice Wilkins, were also actively exploring this field. Pauling had discovered the secondary structure of proteins using X-ray crystallography. X-ray crystallography is a method for investigating molecular structure by observing the patterns formed by X-rays shot through a crystal of the substance. The patterns give important information about the structure of the molecule of interest. In Wilkins' lab, researcher Rosalind Franklin was using X-ray crystallography to understand the structure of DNA. Watson and Crick were able to piece together the puzzle of the DNA molecule using Franklin's data. Watson and Crick also had pieces of information available from other researchers, such as Chargaff's rules. Chargaff had shown that of the 4 kinds of monomers, nucleotides, present in a DNA molecule, 2 types were always present in equal amounts, and the remaining 2 types were also always present in equal amounts. This meant that they were always paired in some way. In 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Medicine for their work in determining the structure of DNA. Figure 9.2. Scientist Rosalind Franklin discovered the X-ray diffraction pattern of DNA, which helped to elucidate its double helix structure. Now, let's consider the structure of the 2 types of nucleic acids, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. The building blocks of DNA are nucleotides, which are made up of 3 parts: a deoxyribose, a 5-carbon sugar, a phosphate group, and a nitrogenous base. There are 4 types of nitrogenous bases in DNA. Adenine, A, and guanine, G, are double-ringed purines, and cytosine, C, and thymine, T, are smaller single-ringed pyrimidines. The nucleotide is named according to the nitrogenous base it contains. Figure 9.3. Each DNA molecule is made up of a sugar, a phosphate group, and a base. Cytosine and thymine are pyrimidines. Guanine and adenine are purines. The phosphate group of one nucleotide bonds covalently with the sugar molecule of the next nucleotide, and so on, forming a long polymer of nucleotide monomers. The sugar phosphate groups line up in a backbone for each single strand of DNA, and the nucleotide bases stick out from this backbone. The carbon atoms of the 5-carbon sugar are numbered clockwise from the oxygen as 1', 2', 3', 4', and 5'. 1' is read as 1'. The phosphate group is attached to the 5' carbon of one nucleotide and the 3' carbon of the next nucleotide. In its natural state, each DNA molecule is actually composed of 2 single strands held together along their length with hydrogen bonds between the bases. Watson and Crick proposed that the DNA is made up of 2 strands that are twisted around each other to form a right-handed helix, called a double helix. Base pairing takes place between a purine and pyrimidine. Namely, A pairs with T and G pairs with C. In other words, adenine and thymine are complementary base pairs, and cytosine and guanine are also complementary base pairs. This is the basis for Chargaff's rule. Because of their complementarity, there is as much adenine as thymine in a DNA molecule, and as much guanine as cytosine. Adenine and thymine are connected by 2 hydrogen bonds, and cytosine and guanine are connected by 3 hydrogen bonds. The two strands are antiparallel in nature. That is, one strand will have a 3' carbon of the sugar in the upward position, whereas the other strand will have the 5' carbon in the upward position. The diameter of the DNA double helix is uniform throughout because a purine with 2 rings always pairs with a pyrimidine, 1 ring, and their combined lengths are always equal. Figure 9.4. DNA, A, forms a double-stranded helix, and in B, adenine pairs with thymine and cytosine pairs with guanine. The structure of RNA. There is a second nucleic acid in all cells called ribonucleic acid, or RNA. Like DNA, RNA is a polymer of nucleotides. Each of the nucleotides in RNA is made up of a nitrogenous base, a 5-carbon sugar, and a phosphate group. In the case of RNA, the 5-carbon sugar is ribose, not deoxyribose. Ribose has a hydroxyl group at the 2' carbon, unlike deoxyribose, which has only a hydrogen atom. Figure 9.5. The difference between the ribose found in RNA and the deoxyribose found in DNA is that ribose has a hydroxyl group at the 2' carbon. RNA nucleotides contain the nitrogenous bases adenine, cytosine, and guanine. However, they do not contain thymine, which is instead replaced by uracil, symbolized by a U. RNA exists as a single-stranded molecule rather than a double-stranded helix. Molecular biologists have named several kinds of RNA on the basis of their function. These include messenger RNA, or mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA, molecules that are involved in the production of proteins from the DNA code. How DNA is arranged in the cell. DNA is a working molecule. It must be replicated when a cell is ready to divide, and it must be read to produce the molecules, such as proteins, to carry out the functions of the cell. For this reason, the DNA is protected and packaged in very specific ways. In addition, DNA molecules can be very long. Stretched end to end, the DNA molecules in a single human cell would come to a length of about 2 meters. Thus, the DNA for a cell must be packaged in a very ordered way to fit and function within a structure, the cell, that is not visible to the naked eye. The chromosomes of prokaryotes are much simpler than those of eukaryotes in many of their features. Most prokaryotes contain a single circular chromosome that is found in an area in the cytoplasm called the nucleoid. Figure 9.6. A eukaryote contains a well-defined nucleus, whereas in prokaryotes, the chromosome lies in the cytoplasm in an area called the nucleoid. The size of the genome in one of the most well-studied prokaryotes, Escherichia coli, is 4.6 million base pairs, which would extend a distance of about 1.6 millimeters if stretched out. So how does this fit inside a small bacterial cell? The DNA is twisted beyond the double helix in what is known as supercoiling. Some proteins are known to be involved in the supercoiling. Other proteins and enzymes help in maintaining the supercoiled structure. Eukaryotes, whose chromosomes each consist of a linear DNA molecule, employ a different type of packing strategy to fit their DNA inside the nucleus. Before the structure of DNA was even uncovered, Marie Maynard Daly and Arthur E. Mirsky conducted extensive research in the 1940s and 1950s to understand the molecules and structures involved. At the most basic level, DNA is wrapped around proteins known as histones to form structures called nucleosomes. The DNA is wrapped tightly around the histone core. The nucleosome is linked to the next one by a short strand of DNA that is free of histones. This is also known as the beads on a string structure. The nucleosomes are the beads, and the short lengths of DNA between them are the string. The nucleosomes, with their DNA coiled around them, stack compactly onto each other to form a 30 nanometer wide fiber. This fiber is further coiled into a thicker and more compact structure. At the metaphase stage of mitosis, when the chromosomes are lined up in the center of the cell, the chromosomes are at their most compacted. They are approximately 700 nanometers in width and are found in association with scaffold proteins. In interphase, the phase of the cell cycle between mitosis, at which the chromosomes are decondensed, eukaryotic chromosomes have two distinct regions that can be distinguished by staining. There is a tightly packaged region that stains darkly, and a less dense region. The darkly staining regions usually contain genes that are not active and are found in the regions of the centromere and telomeres. The lightly staining regions usually contain genes that are active, with DNA packaged around nucleosomes, but not further compacted. Figure 9.7. These figures illustrate the compaction of the eukaryotic chromosome.